Okay, guys, so we're going to talk about section 12.4. This is the last section in this chapter, and it's talking about gene regulation and mutation. And so we're going to finish the chapter up today, and then these notes are actually already posted, and then I'll post your test tomorrow. So it's open note and open book. The only thing I ask is that you just don't give answers to each other. So when we're talking about gene regulation and mutation, the main idea of this is that gene expression is regulated by the cell, but mutations can affect that expression, right? So we're going to talk about gene regulation first. Now, if you remember back when we talked about the different types of cells, there were prokaryotic and eukaryotic cells. And prokaryotic cells were the less complex of the two. Remember, they were single-celled organisms, were made up of prokaryotic cells. There's no organelles, so the DNA is just floating around in the nucleus. But first, the definition of gene regulation is the organism's ability to control which genes are transcribed in response to the environment. So everything that we do is in response. We as humans, we respond to either some type of external or internal stimuli. And so gene regulation is an organism's ability to control which genes are transcribed in response to their environment. So recognize the word transcription, right? That's what we talked about last section. Transcription is the process that gets us from DNA to RNA. So a big part of gene regulation is something called an operon. And an operon is what often controls the transcription of genes. An operon is a section of DNA that contains genes for the proteins needed for the metabolic pathway. So if you remember, we're trying to get from DNA to RNA to protein. So what happens is this process here, this process here is getting us from DNA to RNA, that's transcription. The process getting us from RNA to protein is translation. So notice we're talking about a section of DNA here, and we see that we're foreshadowing the fact that I'm trying to get genes, or I have specific genes that get me to this protein, which is my end result, right? So there should be a few words here that you recognize from previous sections. So there are parts of an operon. There first is an operator, and an operator is a segment of DNA that acts as an on-off switch for transcription. So, when you think about it, if I am an operator of something, I control when it turns on and when it turns off. So that's exactly what an operator does of an operon. It's this segment of that DNA that controls, it's the on-off switch for transcription. Then we have the promoter. If you remember when we talked about transcription, the first thing that had to happen, there was a start sequence, and that start sequence was called a promoter. So specifically, the promoter is a segment of DNA where the RNA polymerase first binds to the DNA. Then we have regulatory gene, and we have genes coding for proteins. So to talk about a specific type of operon, there's something called a trip operon. Now, trip operon is found in E. coli, and E. coli is a type of bacteria. Now, remember, we're talking about prokaryote gene regulation, right? So it makes sense that we'd be talking about bacteria since that is made up of prokaryotic cells. But this is a group of genes that encode biosynthetic enzymes for the amino acid tryptophan. Now, here's when things can get a little bit confusing. The trip operon is expressed or turned on when tryptophan levels are low, and they're repressed or turned off when they're high. So think about it in a way, if I'm in a room, right, and I have a fan in that room, or I have a heater in that room, if the room is hot, I don't have to turn the heater on because my heat level is high enough. But if the room is cold, my heat levels are low, and so I have to turn my heater on. That's the same idea with the trip operon. If tryptophan levels are low, I turn the trip operon on. If tryptophan levels are high, I don't need it, so it's turned off. What kind of goes hand in hand with that is that there's something called a trip repressor, and that is what regulates the trip operon. It's what determines whether or not it'll be turned on or off. And what happens is, in the presence of tryptophan, right, when tryptophan levels are high, my trip operon is off. But the way that that works is because tryptophan binds to my repressor, activating it and turning off the trip operon. Because a repressor is something, if you repress something, right, you're not allowing it to be expressed, you're not allowing it to be shown or seen. So when tryptophan binds to the repressor, it activates the repressor. And as a result, it turns the trip operon off. So here's a picture of that so you can kind of see. If you notice here, this is the section, this is when trip operon is on. Here's my repressor gene, goes through transcription, goes through translation to get to my protein. And here the repressor is inactive because there's no tryptophan present to bind to it. So as a result, the RNA polymerase can bind to the promoter, which then results in transcription, which then results in translation, giving me those enzymes for tryptophan synthesis. However, if you look here, trip operon is turned off because we see here tryptophan is present and it binds to the repressor, activating the repressor and blocking RNA polymerase from binding to the promoter. Now, dealing with the lac operon, this is the same concept of the trip operon, but it's literally the exact opposite in the way that it works. So the lac operon is only expressed, it's only turned on when lactose is present and glucose is absent. So there are two regulators that turn this on and off. One is the lac repressor, which is the one that we'll talk about the most, but there's also something called CAP, which is an activator protein. But the lac repressor, that's what I want to talk about. So it senses lactose indirectly through its isomer allolactose. So this is the opposite concept in that the repressor is bound when allolactose is present, and that inactivates the repressor, which then in turn turns the lac operon on, which makes sense because the lac operon is expressed or turned on in the presence of lactose. So allolactose has to be present to bind to the repressor, inactivating the repressor. So it's the opposite concept of the trip operon. So you can see here, here's my repressor gene. It's active. There's nothing bound to it because lactose is absent, and my lac operon is turned off when lactose is absent, right? So the repressor is active and it's blocking my RNA polymerase from binding to my promoter. However, when it's turned on, lactose is present, and so the isomer allolactose binds to my repressor, inactivating it and allowing the process to happen. So, talking more now about eukaryotic gene regulation. Remember, eukaryotic cells are more complex than prokaryotic. So it makes sense that these require more elements than just a promoter and an operator like prokaryote gene regulation does. So what these require is something called transcription factors. And essentially what transcription factors do is they ensure that the gene is used at the right time and proteins are made in the right amounts. It's like if we had a factory and we were manufacturing something. We may be manufacturing something all day, all year round, but we still have to have regulations, right? Because if we don't have some type of time regulation and amount regulation, then we would just have so much made all the time. There wouldn't be any control. So transcription factors act as that type of regulator. So there's two main sets. The first one is they form complexes that guide and stabilize the binding of RNA polymerase to a promoter. So let's say here I have my strand of DNA. Okay, this is my strand of DNA here. And I'm going to have a promoter sequence, right? That's what I have to hit in order to begin transcription. So what happens is RNA polymerase has to bind, right? I'm trying to get here with RNA polymerase because RNA polymerase has to bind to my promoter. This is my promoter. This is RNA polymerase. It has to bind to my promoter for transcription to begin. And so what happens is these transcription factors come in and they help guide it, right? Can't hardly see this very well, but I'm making them yellow. And these are acting as the transcription factors because they are guiding my RNA polymerase right into the promoter, which is where I need it. These also include regulatory proteins that help control the rate of transcription. So those are things like activators and repressors. So let's erase this and go on to the next slide. Now, there's something called home box genes, Hox genes for short, and essentially what they do is they control differentiation. Now, when you differentiate something, you are just making it different. You're setting it apart, right? The definition of differentiation is process by which cells become specialized in structure and function. Hox genes act as the blueprint for an organism, essentially. If you've ever seen the blueprint for a home before it's built, it lays out what the house is going to look like. Gives you an idea. It's something for builders to follow so they know how to build the house in the right way. Hox genes are the same way. They're important in determining the body plan of an organism, but they actually go one step further. They not only determine the body plan for an organism, but they also control what body part will develop in a given location. So let's look at this picture. I know that's extremely wordy. Let's look at this picture. Here I have Hox genes, okay? And we're talking about a fly here. But notice that it's color coordinated. I have green, dark blue, purple, orange, light blue, light green, yellow, brown. Those are just my genes. Now I'm going one step further into the embryo, which is part of the developmental process. Here's that same green, blue, purple, orange, light blue, light green, yellow, and brown. Now look at the adult fly. Here's green, blue, purple, orange, light blue, light green, yellow, brown. I have the exact same color. So this gene was the blueprint, right? And just like when you build a home, you have to start with the foundation. So you have your blueprint, then you build the foundation, then you get your final product. Well, that's what's happening here. But what's interesting is this section of the gene not only determined this body part, but it also controls that body part. So this specific section of the gene controls the tongue of the fly. This section of the gene controls the tail of the fly. So they go one step further in that they not only say what it's going to look like, it also determines what it's going to do. Now, there's something called RNA interference. Now, we talked about tRNA, which is transfer RNA, rRNA, which is ribosomal RNA, and mRNA, which is messenger RNA. But this is something called RNAi. And basically what it is, it's small segments of RNA. What happens is this enzyme known as a dicer comes in and cuts small pieces of double-stranded RNA in the cytoplasm, and those small pieces become known as RNAi. So what happens is those small pieces of RNA then bind to something called a protein complex, and that whole complex as a whole, the combination of the protein complex and the RNAi, binds to a segment of mRNA. And what happens is once that has bound, it'll cut the mRNA strand, preventing translation from happening. So notice here, right, when I have this strand of mRNA, I know that I've already gone through transcription. Okay, so now here's this RNAi, that single-stranded, small, it's bound to the protein complex. It's going to come in and bind to this section of mRNA, and as a result, it slices it here, and since it slices it, it prevents any of this from being translated. So now talking about mutations, there are two main types of mutations we're going to talk about. There are point mutations and chromosomal mutations. All mutations are changes in genetic material. And so point mutations, however, are just one change in a nucleotide. So within point mutations, there are two types of substitutions. One of them has no effect, and those are called nonsense mutations. Now, what happens in nonsense mutations is a codon is changed for an amino acid to a stop codon. Now, I know you may be thinking, okay, Ms. Brammer, but that's a change, that's an effect. Yes, it is, but it's not changing anything genetically. All it is just stopping anything else from happening. So that's called a nonsense mutation. Now, a substitution with an effect is called a missense mutation. And what happens here is the DNA code is altered so it codes for the wrong amino acid. So instead of it stopping, it changes the amino acid completely, which then changes genetic material. There are also two point mutations known as deletions and insertions, and they are exactly what they sound like. In a deletion, there's a loss of nucleotides in a DNA sequence, and in an insertion, there's an addition of nucleotides in the DNA sequence. So looking at these a little more closely, this is what we call an example of a substitution with the effect. So missense mutation. If you notice here, here's my strand of DNA, right? I go through transcription to get to translation. I go from DNA to RNA through transcription. Then I go from RNA to my protein or amino acids in translation. So notice in my DNA strand here, I have the base pair C, cytosine. But here, I have T instead. And as a result, that transcribes completely different than what it did here, and then translates completely different. So I have a totally different amino acid here, which changes genetically my outcome. An example of this is sickle cell anemia. And sickle cell anemia is when someone does not make healthy red blood cells. So this is the normal shape of a red blood cell. This is the shape of a red blood cell with someone who has sickle cell anemia. It kind of looks like a red green bean, I think. And then what this picture is just showing you is it's that one amino acid that has changed. So here's an example of an insertion. So if you notice, I have the same amino acid, I have the same nucleotide bases here and here, but there has been a nucleotide base inserted. And as a result, I still have to follow the rule that there are 3 nucleotides per codon. So when this is inserted in the third position, it shifts everything over to the right one spot, which then changes my coding for the amino acids when it's translating. So here I have completely different amino acids than what I do here. And that is a result of an insertion mutation. And finally, I have deletion. Now this clearly is not our nucleotides, but they're trying to make a point showing you that this sentence is completely legible, right? The fat cat ate the rat. That's a sentence that I can read. But when I have a deletion, one whole nucleotide is deleted, and as a result, everything shifts now to the left because I still have to follow the 3 nucleotide rule per codon. And so that's what this is showing you here, is what I started with was a legible sentence, and now I have something that I can't read at all. And an example of this is cystic fibrosis. So we also have something called a chromosome mutation, and these are changes in the entire gene. And I know we've not talked a ton about chromosomes yet, so just let me show you what a chromosome looks like. So they're double-stranded, and they look like an X, basically, and they meet right here in the middle at a spot called the centromere. So, shoot. Keep an eye out for this, because you're going to recognize a chromosome as we talk about these different types of chromosome mutations. So chromosomes are made up of entire genes, several genes, and there are 4 kinds of chromosome mutations. There's inversion, translocation, deletion, and duplication. So starting with inversion, see, this is a chromosome. It's just bent in a funny way because they're trying to show you what happens. But if you see, this part of the chromosome is made up of 3 genes, and this part of the chromosome is made up of 3 genes. So I have A, B, C, and D, E, F. In inversion, literally one gene inverts with another gene from the opposite strand. So instead of having A, B, C, I have A, E, D. Instead of having D, E, F, I have C, B, F. An example of this is Tay-Sachs, which is a lung disease. Now here I have translocation. So here I do, once again, I have this is one strand of DNA, or one strand of my chromosome, another strand of my chromosome. And what happens is it's more than just one gene that changes this time. Instead, I have a whole segment that translocates. It moves across. If you go trans is across, location is spot, so it completely goes across and changes locations. And you can see that here, right? I have ABC, but then I have JKL instead of DEF. And an example of this is leukemia. Now, in deletion, what happens is nothing changes positions and nothing gets replaced. Instead, an entire gene is just deleted. So notice here, I've lost B when I have a deletion. So I have 6 genes on this chromosome, but I have only 5 because I've lost 1 gene. An example of this is Williams syndrome, which is something that I had never heard of before. I hadn't heard of it, and I Googled it, and it's kind of interesting. It's opposite of autism in that some people with autism are very they struggle in social situations and do well socially. However, people with Williams syndrome are extremely outgoing. They still get fixated on things like some people do with autism, but they're very outgoing. But what's interesting with people with Williams syndrome is their facial structures are very unique. There's a deformity in the structure of their face and the bones, and so they have this unique look about them. But that's my extent in Williams syndrome. So if you want to know anything else, you're going to have to look it up. So then here we have duplication, and this is the opposite of what deletion was, right? We are, instead of losing one, we're gaining one. And it's not the same. I don't know if you've ever been writing something really fast, maybe numbering something 1 through 10, and you accidentally write 2 twice instead of writing 3. And but that's not the case. This doesn't replace anything at all. It just duplicates. So I have an additional one. If you notice, I have 6 genes here, but I have 7 genes in this one because I have duplicated my B gene. And some examples of this are muscular dystrophy and Huntington's disease. So what is crazy to me to think about is that we are made up of billions of cells, okay? And genes, chromosomes, make up DNA. So you think about the fact that each individual cell that we cannot see with the naked eye is made up of DNA, and that DNA is made up of chromosomes, and it takes one simple mutation in one of those chromosomes to result in something like this. So, two more slides. There are causes of mutation, and mutagens are the substances that cause mutations, right? There are certain chemicals and radiation that can damage DNA. Like that's why people who have gone through radiation treatments for cancer are more susceptible to other diseases, because it's easier for mutations to occur within their DNA. And that's also why, just as a whole, the general public is asked to limit their radiation exposure. However, going back to the second section of this chapter with DNA replication, DNA polymerase is what unzips and so DNA polymerase will unwind DNA and then go back through and add nucleotides. And sometimes it'll add the wrong nucleotides. However, it's only 1 in 400,000 times that it adds the wrong nucleotides. But after it goes through and adds nucleotides, it also goes back through and proofreads. And so most of the time, it catches that mistake that it did make. It's only less than 1 out of 1 billion times that it goes unfixed. Unfortunately, it's just that less than one time that can be detrimental. But what happens is it affects the DNA by changing the chemical structure of the base. So when I write out a strand of DNA on the board, you just see the nucleotide bases. You see A, T, C, G, T, A, G, C, whatever it is, whatever the order is. But what happens is there's also a chemical structure to that that you cannot see. And the mutation will change that just enough, change the chemical structure just enough where it still is an adenine, but it looks like a cytosine. And so instead of thymine pairing to it like it should, guanine will come in and pair. So it causes bases to mispair or bond with the wrong base. And then finally, there are different types of cells in our body. We have body cells and sex cells. And so mutations in body cells become part of the genetic sequence in that cell and in future daughter cells, but they're not passed on to the next generation. So cancer, for example, let's say that my mom had cancer, that cancer can affect her cells in her body, right? It may start in one cell, but it gets passed on to the daughter cells and it can spread throughout her body, but it won't be passed on to me as her child. It's not genetically passed on to me. Now, am I at a higher risk because she had it? Yes, just because of family history and all those things. But I'm not automatically inheriting it genetically. Sex cells, however, are passed on to the next generation. So you take something like a sex cell, a disease that or mutation in a sex cell like Huntington's, that can be passed on from generation to generation. And unfortunately, sometimes it can be more detrimental in the offspring than what it ever was in the one who first had it. Because, let's say, for an example, my mom and dad both were carriers of Huntington's. They would both pass on that gene to me, and as a result, I would express it. And so, we'll talk more about Punnett squares and genetics and passing on things next chapter. But just know that sex cell mutations can be passed on and can sometimes be more detrimental to the offspring, whereas body cell mutations just is limited to the body of the person that has the mutation.